We go on
Thanks,
to
Mark.
our second speaker, Andy Walker. He's also in Australia. He's in Brisbane at the Institute for Molecular Biosciences at the University of Queensland. He's a research fellow there. And if you're thinking, I would like to meet an arthropod venom specialist, well, then we have Andy here for you. He's worked on a whole bunch of different arthropod venoms, spiders, ants, assassin bugs famously, but also lepidopterans. And it is lepidopteran venoms that he's going to talk to us about today. I see that you managed to get a functional laptop, Andy. Very happy to see that. So
can you see that?
yes, we can see that, and the
Excellent.
digital platform is yours.
All right, thank you so much. Thanks everybody. Gonna wing it a little bit. Had to do a last minute change of laptop, but hopefully it's working. If it's not, please yell out, and we'll see if we can fix it. I'm gonna talk to you today about some venoms that are made by Lepidoptera. So Lepidoptera are, of course, the butterflies and moths, but in their larval stages, they're also caterpillars. So it's the adults as well as the caterpillars that make defensive venoms and toxins. But by far, the caterpillars are the real masters at it. So how many species of venomous leps are there? There might be about 3,500. We estimated in a recent review, and you can sort of think that that's a lot, 3,500, or you can consider it's actually not that many. It's about 2% of the order. If you look at the actual phylogenetic distribution of those venomous species, what you see is they're actually really spread out in this group. So it's a very different distribution than you would see in something like spiders or centipedes or scorpions, where they evolved venom anciently and almost all species retained them. In this instance, they probably arose multiple times, at least 4 times, although we don't really know the right number. But then they've been lost multiple times as well. And that probably relates to them being defensive venoms. So these are venoms made by herbivorous species that are used for predator deterrence. So the introduction that Richard and Ron gave to defensive versus predatory venoms is probably very useful there. So thank you.
And Andy, can I ask you a quick question? There is a little bit at the bottom of your screen that is obscuring some of your images. Could you click on the hide button? Do you see what I mean?
Oh, I see. Yeah, here.
Rock and roll.
Is that better?
Thanks. Yep.
Excellent. Thank you. In terms of what those venoms do when they get into an animal, typically they will cause pain and irritation, but there are actually some pretty weird envenomation syndromes that you get with caterpillars. So there are 2 separate groups whose venom is associated with causing reproductive problems in horses. And there are caterpillars that cause chronic inflammatory conditions. There are caterpillars, one genus of caterpillars whose venom can kill people. This one, for example, is a caterpillar who, if you get envenomated by it, can give you a chronic condition, sort of like rheumatoid arthritis, that can actually be disabling. It can prevent you using your hand ever again. All around the world. Our Australian one has the dubious prize of also causing equine fetal loss syndrome. So a mare accidentally eats one of these caterpillars, or it sheds skin, and it gets these tiny
Oh no.
but a potentially fatal one, which is genus Lanomia, and it's given. The way that venom actually kills people is by causing coagulotoxicity. So it's promoting clotting in the body, but then that's going to interact with feedback networks, and what you actually get is unclottable blood and a hemorrhagic syndrome, and you can die from brain hemorrhage or kidney failure. So there are all these different groups of venomous caterpillars and a few groups of venomous. A little bit about a couple of these families. Starting with the Lemurcotidae, which is where we started looking at caterpillar venoms. And the Lemurcotidae are part of this superfamily called Zygonoidea. And quite a few of the families in this superfamily have some pretty interesting defensive adaptations. So we have the Zyganidae, which have these little turrets that produce noise. We have the asp caterpillars or toxic toupees. These are venomous caterpillars that have a really painful venom. We have the Dalceridae that are called jewel caterpillars because they're covered in these kind of beautiful adhesive droplets that have a defensive function. And then we have the Lemurcotidae, about half of whom don't have venom, and they're known as jellies because they look like a cute little jelly bean. And we have the nettle caterpillars, which are the ones with these pain-causing venomous spines. Here's a little video for you for the species, the first species we started working on. It's called Doratifera vulnerans, which is a binomial name, which, as far as I can tell, translates as bearer of gifts of wounds. That has venomous spines that are actually retracted at rest, and when it thinks it's being harassed, it can get those out. Presumably, it does that because it doesn't want to something. So we can look at the composition of these Lepidopteran venoms using transcriptomics and proteomics, and we do that by harvesting venom from the tips of the spines just by pressing some parafilm on it. That ensures that we don't get any housekeeping and cellular proteins like we would if we were taking an extract from those spines. And we can do a transcriptome of the spines and a little bit of the surrounding tissue, which we know, won't go into it here, but we know is where that venom is being produced. Doratifera venoms typically really rich in peptides, like you can see over on the left. There's quite a few different families present, and there's one that's derived from arthropod neuropeptides, and then the second one that are these cationic amphipathic peptides, which reminded us of the venoms, some of the venom toxins produced by ants and bees and wasps, but which we think have evolved from an immune peptide in non-venomous lepidopterans called cecropin. And then there's disulfide-rich peptides there as well, which are eerily reminiscent of the peptides that dominate spider venom. And they probably fold into this inhibitor cysteine knot or knottin structure. We did this in two ways.
Andy, can you hear me? I think the connection to where you are is pretty poor. I'm thinking it is your end, and not my end.
Both of them take.
Can other people hear Andy as well, or did we just completely lose him?
I think we lost him.
I think we lost him. Okay, so that
Yeah,
it
we lost
confirms
him.
it confirms it's not our end. It is there.
Oh.
Andy is again. You fell out, Andy.
I did.
Yes, so it was quite hiccupy the last. It was quite hiccupy the last couple of minutes, and then you just fell out.
Maybe since I showed the video.
It was already a bit hiccupy before, but the video didn't help.
Okay. Shall we just keep on, or
Yeah.
what do you want to do?
So could you the slides immediately after the video with the two pie charts? That was literally up for half a second because it was trying to catch up. If you can start there again, is that possible?
Yep. Can you see my presentation?
Yep, I can see your presentation. I can see Sam Robinson.
Okay. You can see Sam Robinson.
Yeah,
So
I
I've got
can
the.
see. I see a slide with neuronal activation.
Oh, that's so weird. I've got a different one up. I've got the pie chart. Let me just check it hasn't done something weird with the
Yep,
internet.
I can see the pie charts now.
Yeah. Just gonna check which internet it's trying to use. Yep, it's the right one.
You know what could help if you turn your well, you can't turn your video off, can you? No.
Well, I can turn it off and yeah.
Because maybe it's because we can see your talking. Yeah, maybe this helps because then it keeps some more broadband available.
Okay. So if I do this. Okay, can you see that?
Yep. I mean, we
pie
can.
Charts?
Yeah. Let's see. It's not in presentation mode yet.
Okay. Just give it a second. It sounds like there's a bit of a lag in the visuals compared to the audio.
Yeah, I think you're right. Still waiting.
So it still doesn't look like presenter mode?
No, it's still in browsing mode with the little slide thumbnails on the left.
Okay. Sorry about these technical issues.
Oh, it happens. It's
I
not your
could do
fault.
this from home, but I might need to do it from the lab next time, I think. I'll just try stopping sharing and then sharing again.
Yeah, maybe that's a good idea. I think we are in business.
Okay, you can see my pie charts?
Perfect.
Okay, hopefully we
that.
won't have more problems, but just yell out if we do. So there are quite a few different families present here. There were three that were particularly abundant in the venom. The top one there is the family that we think is derived from arthropod neuropeptides. Then the second one in the middle are the family of cationic amphipathic peptides, which are similar to ant and bee and wasp toxins. And then the third one there are the inhibitor cysteine, not like disulfide-rich peptides. Then this is the assay we used to look at what was causing pain. So this assay, you get the pain sensing neurons, peripheral sensory neurons. You fill them with a calcium sensitive dye, and then if you're applying a stimulus that's likely to cause pain, then what you expect to see is these cells light up in fluorescence, and that's graphed over on the right there, where each little grey line represents a single cell, and then the blue lines are showing you averages and interquartile ranges. So we can apply the venom by itself or in the presence of tetrodotoxin or divalent cations. And when we do that, we see that the venom still works fine. So we think this is telling us it's not working by voltage-gated sodium or voltage-gated calcium channels. But we also took that venom and we fractionated it using HPLC. And then we applied each of those separate peptide fractions in this assay. We found the ones that had the strongest response. We used mass spec to identify the toxins, and then we synthesized them, and they really nicely recapitulated the action of the whole venom, including not being inhibited by these pharmacological agents. And we think that all makes sense because these peptides are members of that cationic amphipathic family of peptides, which are known to permeabilize cell membranes directly. So what about this this next family over, the Megalopigidae or asp caterpillars? Will they have venom that acts in a similar way? I'm just going to skip this because it has a video in it. So we had a look using proteomics and transcriptomics, and what we found is that the venom is entirely different. So yes, there are linear peptides there. Yes, there are disulfide-rich peptides there. But they're from completely different families to the ones that we saw in the lemur-coated venoms. And beyond just the peptides, the thing that jumped out at us is that there were a whole lot of proteins there, larger proteins, around 30 kilodaltons, which when we annotated them, they told us they were bacterial toxins, specifically from this aerolizin family. So they're actually homologous to the crystal or cryotoxins that we use to protect crops from insects in for BT cotton and other BT crops. We can apply those venoms in Sam's assay again, and we see that they really nicely light up all the cells there. And at the time we did this, I figured it was going to be some of those peptides present in the venom. So we went ahead and we made them all and we applied them in the assay, and it didn't actually, none of them actually caused a strong response. So then we tried to make some of the larger proteins using recombinant expression. That also didn't really work. So we ended up doing this to try and work out what component of the venom was responsible for causing pain. And what it is we took some venom. This is a lower concentration, so it takes a little bit slower to onset in this assay. And we treated it with acetonitrile or with heat or with proteases, and just had a look at what that does. And you can see in each case it basically wipes out the activity of the venom. So just adding 50% acetonitrile, waiting for 5 minutes, drying that down, and resuspending it in water just kills the activity, as does a mild heat treatment or treatment with a protease. So this is telling us that the toxins responsible are larger toxins than peptides, which you would expect to survive at least 2 out of these 3 treatments. And it's not a small molecule, which would survive all of them. It's a larger protein. We did that in vivo as well. You can apply the venom and see this strong increase in pain-like behavior, but it's wiped out if you pre-treat that venom with acetonitrile and then dry it down and resuspend it. You can, we did a few other things to have a look at those toxins, the megaliazins. You can chuck them in AlphaFold and see what it thinks their structure is. And if you do that, it will predict that they are almost exactly the same as the monomeric forms of these bacterial pore-forming toxins. So these toxins take up a really different structure when they assemble on a plasma membrane. They turn into this giant beta barrel mushroom tree thing that you can see in the top right. And there's certain sequence features that we think enable them to do that. And one of those sequence features is these alternating hydrophobic and hydrophilic residues that you see in the parts that are eventually going to form the pore. That's because these this region, the side chains have to stick out. So alternating side chains will stick out on opposite sides of the sheet, and one is going to be in the aqueous pore, and the other one is going to be in the membrane. And we see that our caterpillar toxins retain all those features, so they seem like it's consistent with them being pore-forming toxins. We've also looked at how they evolved. So the most similar thing you can find to these venom toxins are proteins of unknown function that are made by a non-venomous Lepidoptera. But if you look outside of that group, you see that the most similar proteins are actually among bacteria. We went ahead and did some phylogenetics to look at that in more detail. And this is what we ended up with. So the megalysins are up in one clade. They're all monophyletic. So I think that's telling us that they've been recruited as venom toxins just once. And then they're housed within this larger clade that's all the Lepidopteran sequences we included in this analysis. And that's also monophyletic. And we think that that means that there was one lateral transfer event. Then the sister to that clade is gamma proteobacteria. So I thought that this was really weird. Turns out it's less weird than I thought. There's been horizontal transfer of aerolysins into from bacteria to a pretty diverse range of organisms, and we think this has happened about 8 or 9 times based on the data we have so far. So just to sum up, we think it's those larger peptides, larger proteins that are related to bacterial pore-forming toxins rather than the peptides that cause pain. This shows that horizontal gene transfer contributes to venom evolution, but the other reason I thought it was neat is that it's the first example of a caterpillar using a cryotoxin against a human, which is the opposite of what happens when we protect BT crops from caterpillars. I'm aware that because of the technical issues, we're a little bit behind on time. So, Ron, if you want me to stop, just sing out, and I'm happy to stop anywhere.
No, I think people can leave if they need to go to the next meeting. So I would like you to continue. No worries.
Sure. Okay. So, at the time we did this, we thought, well, the venom's completely different. We also looked at the anatomy to do with venom production, and that was different. And we thought, you know what, these two families of caterpillars have just evolved venom completely independently. And as always, when you have a nice, clear result in science, you get some more data, and it kind of knocks your theory apart. So I'll finish with these two stories that have come from the PhD of Desi Gudazi, who's just finishing up now. Great student, will make a great postdoc. I think she's looking for jobs now, if anyone's in the market. So the first one I'll tell you about is this saddleback caterpillar that we actually saw in one of Richard's slides. And the second one is from this one on the right that we call the electric caterpillar. So we wanted to look at some more diverse species, and we decided to pick one that was far away. So that was the saddleback caterpillar from North America, and then one that looked kind of different. At the time, we didn't have a good phylogeny of these caterpillars, but we resolved that in the process. We were surprised when we looked at the saddleback caterpillar. The venom looked really, really similar. So most of the same peptide and protein families were represented. And we could even align them, and some of them had very few differences. Some of them had more. But one thing we found in the saddleback we didn't find in the Australian species Doratifera vulnerans was this RF amide family of peptides. So if you haven't come across these peptides that end in an arginine, phenylalanine, and then an amidated C terminus, they're really widespread across the tree of life, used as endogenous signaling molecules, and they're typically the ligands of GPCR receptors. Desi happened to notice that one of ours, AS54, was actually identical to one of the human endogenous signaling molecules in the last 4 residues. And so David made some of these peptides and Desi purified them, and then we gave them to James, who tested them in a Presto Tango reporter system for this GPCR. And what he found is that it's actually equipotent with the human endogenous ligand. And those peptides were also insecticidal as well. So we aren't really sure, but potentially these peptides may be protecting the caterpillars from arthropod as well as vertebrate predators. So the last story here is just about this guy, the electric caterpillar. When we started, we didn't have a species name for it. We ended up doing some DNA barcoding, which is something you can do as long as people have been collecting moths and sequencing them. And the databases there for moths are actually really good now. So when we did our venom gland transcriptome, we searched for CO1 barcode, got it out, put it in the database, and it will tell us what species it is. And the species is called Camana monomorpha. When we went to collect these things, we didn't really know where to find them. So I thought I'd be clever and go on a Townsville gardening Facebook group and post a photo and ask if anyone had seen them. And we really weren't prepared for the outpouring of wrath and hatred that gardeners in Townsville felt for this caterpillar. We heard about people who had been to hospital multiple times with anaphylaxis-like syndromes, which we hadn't heard about. We heard about people who had removed entire trees or hedges to try and get rid of them. And we were lucky enough with Michaela Mitchell to record a detailed case study showing that the lesions you get from these caterpillars can last up to 2 weeks. And lo and behold, when Desi did the proteotranscriptomics, we found their venom was very different from the other lemicodids that we had analyzed. So instead of having all of these peptides and ICKs, we found very few of those. Instead, we found a whole lot of aerolizin-like toxins. And you can see this in the SDS page up on the top left. The most abundant thing there are these pore-forming toxins. And that venom beautifully permeabilizes cells, just like you would expect. We can see that in Sam's assay, which is shown at the top, and also this assay that Fernanda developed, which is sort of cool because it directly measures membrane permeabilization by measuring the exposure of DNA and RNA, which should be inside the cell, to propidium iodide that you'll apply outside. And they were super potent. So the IC50s here are around 1 microgram a mil. Now, is this just because Doratifera and the saddleback Acaria are more closely related to each other than they are to our spiky Camana monomorpha? Turns out it isn't. Both of the Australian species are more closely related to each other than they are to the American saddleback. So this leaves us with a few questions about how venom evolution has proceeded in this family, but we've actually got about 8 more data points coming through now, which is giving us a bit more of an idea. So in summary, we have these caterpillars in family Lemicodidae. They tend to produce venom-rich peptides, including these membrane permeabilizing amphipathic toxins. We have the megalopygid caterpillars, whose venom contains aerolizin-like toxins. We looked at the RF amide peptides of the saddleback caterpillar and how they're insecticidal and activate human GPCRs. And lastly, we saw the electric caterpillar that shows that lemicodids also can have aerolizin-like toxins dominating their venom, and suggest possibly the 2 families are descended from a common venomous ancestor. So lastly, I'd just like to say thank you for everyone. If you're still listening, Thank you to the organisers for inviting me, and thanks to everyone who took part in the project.
Thank you, Andy. This is the first time I'm seeing a acknowledgement slide that explicitly says, "Thank you to the people I've forgotten." Sorry about that.
Yeah, I just thought I'd be preemptive on that.
Very thoughtful. People, comments and questions, this is your chance. You can type them in the chat, or you can put your camera on and speak up. I have a question. Are you familiar with the so-called vampire moths in the genus Calyptra?
Yes.
Would you consider those venomous?
I would consider anything that uses a saliva or another secretion to facilitate blood feeding venomous, because I like that really broad definition. And we know a lot of them have, multiple or species dependent effects. So I think a broad definition is better for researchers anyway.
Yes.
We have tried looking at them a little bit, but we had a lot of trouble getting biological material. The species we wanted to collect the most were in Siberia. I think we had a transcriptome. This was a collaboration we started with a researcher called Zaspel, but it never got off the ground because we could never get any pure saliva.
Yeah. And it's also, I mean, I looked a little bit at the literature for a talk I gave at the IST meeting recently. And they seem to salivate copiously, but nobody really knows if that saliva is used in any functional way. So it's sort of a blinking question mark. So yeah, it would be awesome if you would manage to at some point look into that.
That would be great. Yeah.
But that's in that sense, if you just do research like I sometimes do, which means you read the literature, that's my exposure to the empirical data. You sometimes find other groups like velvet worms and onychophorans. By the definitions we use, they may be venomous too, because the only study that's been published on their behavior, the predator behavior, yes, they use their sticky slime to immobilize initially prey, but it's the bite they give and the saliva they inject that kills them quickly and
Is it really?
Yes.
That's fascinating. Yeah, I didn't know that. I've seen them and I've collected them and I'm aware of work on the glue. But I mean, if that's true, they're maybe a bit like spiders. They've got a bioadhesive, but also the venom as well.
Yep, exactly. So no, so and if you look at the literature, there's some other cases where, I mean, these are not new categories of venomous creatures. They're still, I guess, perhaps another data point in the roster of predatory venoms, but still, that's cool.
It is cool.
People, any other caterpillar or Lepidoptera based comments? Nice talk, Andy Blarina says.
Hey, Andy. I just wanted to say I don't
nice to see you.
Yeah, nice to see you as well. I just wanted to see a great presentation. I really enjoyed it. I don't have a question in particular, but I still really liked the presentation.
Thank you. Cheers.
It was striking, the horizontal gene transfer thing. I mean, more and more it seems like, and of course we know that horizontal gene transfer has helped build adaptations in eukaryotes across many different sort of organ systems. But in venom, it seems more and more, especially in the invertebrates we love. It's quite a powerful force, which is interesting.
It's amazing how much of it is going on all the time. And I mean, you don't think about it, but the world's kind of a sea of DNA, which is sloshing around with organisms moving through it. It's possible and likely that it does get incorporated into genomes.
This is one of the reasons why I've got a PhD student, Harriet, who's in the chat as well, who was looking at the microbiomes of centipedes to try to get some understanding of, the sort of reservoir and pool of available microbes. Where are they? Because the mechanisms of horizontal gene transfer in eukaryotes are really poorly understood.
Totally. Yeah. I mean, I always think it's an interesting coincidence that, I mean, you have these pathogenic bacteria with pore-forming toxins that are, you know, a big part of their pathogenicity, that, you know, are opening up cell membranes. So, I mean, is it invertebrates that are being attacked by these things who end up, you know, getting the horizontal gene transfer, and it does the DNA get in through the pores?
Yeah.
It's a lot to think about. I don't know how you would actually
Yep.
test something like that.